Hello dear viewer, welcome to my YouTube channel. In this video lecture, the topic under discussion is nucleic acids. There are two types of nucleic acid. One is DNA and next is RNA. The DNA and RNA are collectively called nucleic acid, and the basic units of nucleic acid is called nucleotides, which are adenine, guanine, cytosine, thymine, and uracil are in case of RNA. Components of nucleotide. A single nucleotide have three components, that is pentose sugar, which are two types, that is ribose and deoxyribose sugar. Ribose sugar are present in RNA while deoxyribose sugar are present in DNA. A phosphate group and nitrogenous base. Phosphate group, which are phosphoric acid made by 4 oxygen atoms and are negatively charged, and nitrogenous base are two, that is purine and pyrimidine. Now let's discuss each component in detail. Now the pentose sugar of nucleotide, which are two types, that is deoxyribose sugar present in DNA and ribose sugar found in RNA. In DNA, this is the deoxyribose sugar, which are 5 carbon sugar and hydrogen are present at position 2, while in RNA at position 2 hydroxyl group are present. These both are 5 carbon sugar, that's why it's called pentose sugar. Next is nitrogenous base. Nitrogenous base are purines and pyrimidines, where the purines are double ring structure, that is guanine and adenine. These nucleotides are found in both DNA and RNA. Minor purines are xanthine and hypoxanthine, which are not mentioned here. Pyrimidine are single ring structure. They include cytosine, thymine, and uracil, where uracil are present in case of RNA only instead of thymine. The cytosine are found in both DNA and RNA. Thymine are found in DNA and uracil are found only in RNA. Now have a look at the structure of purine and pyrimidine. Purines are two, that is guanine and adenine. The guanine are double ring structure, and in biochemistry we called it 2-amino 6-oxypurine, because in position 6 oxygen are present and amino group are present at position 2 or carbon number 2. Now adenine are also double ring structure. They are called 6-amino purine in biochemistry because in position 6 or carbon number 6 amino group are present. Pyrimidines are cytosine, thymine, and uracil. Cytosine are single ring structure, not just like purine, because they are double ring structure, and we called it 2-oxy-4-amino pyrimidine in biochemistry because oxygen are present in position 2 or carbon number 2 and carbon number 4 contain amino group. Next is thymine. Thymine are denoted by T and present in DNA only. In biochemistry we called it 2,4-dioxy-5-methyl pyrimidine, or it's also called 5-methyl or 5-methyl uracil because of its structural similarity with uracil. The uracil have no methyl group while thymine have methyl group. At position 5 or carbon number 5, uracil are also single ring structure and are present instead of thymine in RNA. In biochemistry, we called it 2,4-dioxypyrimidine because in carbon number 2 and in carbon number 4 oxygen are present. Next is phosphate group. The phosphate group consists of phosphate, which is phosphoric acid, and are bounded to 4 oxygen atoms, and their one oxygen is bounded with carbon number 5 of ribose sugar. This is the structure of triphosphate because 3 phosphate groups are present. Now we must know what is the difference between nucleoside and nucleotide. The nucleoside is the nitrogenous base plus pentose sugar. When the phosphate group is added to this nucleoside, then we collectively called it nucleotide. This is the structure of nucleoside, and when we add phosphate group to the nucleoside, then we called it nucleotide. The nitrogen base bond with pentose sugar. Carbon number 1 of pentose sugar bond with N1 of pyrimidine by BN glycosidic bond. The carbon of sugar are denoted by prime to separate it from the carbon of nitrogenous bases. The carbon number 1 of pentose sugar and N9 of purine by BN glycosidic bond. The bond that attach pentose sugar to phosphate group is called phosphodiester bond. Now the suffix used for purines and pyrimidines. As we know, purine are double ring structure. They will have suffix sign, and pyrimidines are single ring structure and they will have suffix dine. For example, if the nucleoside containing pyrimidine, they will have suffix thymidine, uridine or cytidine, and if the nucleoside containing purine, they will have suffix sign like adenosine, guanosine, etc., etc. Thanks dear student for watching. If you like the video, like, share and comment.